Hi everyone, welcome back for lesson 4.3. We are continuing with Punnett squares and probability, but today we will be doing dihybrid crosses. So if you did not get to finish 4.2, the video, the notes, or you've had kind of a hard time with 4.2, make sure to ask questions with your teacher before moving on to this lesson. So a couple things that we're going to review before we start this lesson. If you remember last time, we talked about these terms homozygous, heterozygous, genotype, and phenotype, okay? So homozygous are organisms that have two identical alleles for a gene. So for instance, big T, or little t, both of those are homozygous. This one, however, is homozygous dominant. This one would be homozygous recessive. Heterozygous is organisms that have two different alleles for a gene. So a big T and a little t would be two different alleles, okay, and that would be heterozygous. Now the heterozygous is always going to look dominant, okay, or is going to kind of look like the homozygous dominant one, however their genotype is different. The genotype, again, is the genetic makeup, so literally just the letters or the alleles that make it up, and the phenotype are the physical characteristics, so in this case, physical would be tall or short. So also a review from last time, this terminology, we had our P generation, that was the parental generation. Remember that was that true breeding, okay? If you see true breeding, again, you're thinking P. F1 was the offspring of the P cross, okay, the F1 generation, first filial generation, and the F2 was the second filial generation, and it's the offspring from the F1. Now, Mendel's experiments, if you recall, the F1 cross was a cross as a result of the 2P generation, two true breeding plants here, and they all ended up looking like the P generation, okay, or one of the parents from the P generation, right? So tall, axial, green, smooth, all of this F1 offspring ended up looking like this parent in particular. Now, why is that important? Because when we talk about our F2 generation, then we came up with this law of segregation. So remember, during gamete formation, the alleles segregate from each other so that each gamete carries only a single copy of each gene. So here's kind of a good demonstration of that. Also, whenever you do monohybrid crosses from our last lesson, this is essentially exactly what you do, right? You separate the gametes, you put them in two separate boxes at the top and the sides of your Punnett squares, and that gives you four different unique combinations. Now, moving on, okay, independent assortment is a new concept. So, after showing that the alleles segregate during the formation of gametes, Mendel wondered if the segregation of one pair of alleles affects another pair. So, for instance, does the gene that determines the shape of a seed affect the gene for seed color? And so, to find out, Mendel followed two different genes as they pass from one generation to the next. So, in this case, if we look here at this picture, he's looking specifically at color, okay, yellow or green, and seed shape, which was round or wrinkled, or smooth or wrinkled. So for F1, K. Mendel crossed true breeding plants with only round yellow peas, and those produced only wrinkled green peas, and all of the F1 offspring had yellow peas. However, in the F2, that's when we start to look a little bit different. So the F2 generation, the pea plants exhibited approximately a 9 to 3 to 3 to 1 ratio, as seen in the Punnett square. And we're going to see this ratio come up quite a bit. So independent assortment is the genes for different traits can segregate independently during the formation of gametes. So what's really the big difference between segregation and independent assortment? So one way you can kind of think of this is segregation's mostly looking at things like monohybrid crosses. Okay, independent assortment is looking at what we call dihybrid crosses, or whenever we are looking at two genes or two alleles or two different things. Okay, so for segregation, it's the two alleles for each gene separate during gamete formation, and for independent assortment is the alleles of genes on non-homologous chromosomes assort independently during gamete formation. Okay. Okay, so we're going to go ahead and look at some practice problems. Okay, so for our first one, we had two pea plants and they are both heterozygous for size and pod color, making their genotypes here, heterozygous. Okay, big T, big G, little g. So for our first step, we're going to determine the gametes that each parent can make, and each gamete should have an allele for both genes. So in this case, what we kind of do for this is similar to what we do in math whenever we do FOIL. I'm not sure if you guys remember FOIL, but we're going to do something kind of similar. So the genes, okay, the gametes that the parents can make will look like this. This T could go with this G, and it could make that possible gamete, okay? This T could go with this G, that's our second combination. Okay, this T could go with this G. That is your third possible gamete. okay, little t, big G, and our last one, this T could go with this G, and we get those, okay? So when looking at dihybrid crosses, our first step is just to look at the possible gametes the parents can make. Okay, they can make 4 possible gametes, and here, because both parents are both heterozygous the exact same, this will be the possible gametes that they can make. All right, so step 1's here. Okay, step 2, we're gonna make, since each parent can make 4 types, we're gonna make a 16 square Punnett square. Okay, so we're gonna go ahead and put that. And then we're going to put the 4 possible gametes along the top and the sides. And for this particular problem, since both parents have the same genotype, they will look the same. So we're going to go ahead and put those gametes. Again, we just got those here, okay, on the top and the side, okay. Now, step 3. Okay, this is a little bit more of the tedious aspect, but we're going to fill in the table by combining the gametes' genotypes. When filling in the Punnett square, always put your capital letters first and group the same letters together. Now, what does that mean? Well, here's your first line. So for instance, I'm always going to put big T, okay, big G, big G. So I'm going to keep my T's first, G's second. Here, same thing, big T, but now we have big G, little g. And so why do we want to put the capital letters first? Because that's kind of how we understand whenever we see heterozygous things, right? We want to have big G, then little g. Okay, and then we have this line and this line. So now that I've done this first line for you, I want you to pause here and go ahead and try to fill in the rest of this Punnett square. Okay, so we should have gotten something that looks like this. Now again, always keep your letters together. So T's first, G second, capital letters go first, lowercase letters go second. Okay, so we should get something like this. Now, as you're looking at this, you might be thinking, well, what do those different colors mean? So our last step for this will be to determine the phenotypic ratio, okay? Now, the phenotypic ratio is here, okay? It's in colors, but we go number of dominant for both traits, dominant for first but recessive for second. Our green is the recessive for the first trait but dominant for the second, and the purple is where we have recessive for both traits, okay? So basically, this red, dominant for both, would mean that we have to have a big T and a big G, okay? If we have that, it would be red or the first thing in that phenotypic ratio. Second thing, dominant for the first trait, meaning a big T, and recessive for the second, meaning a little g, okay, or both have to be little g's. Third one, recessive for the first, meaning has to be 2 little t's, okay, and then we have dominant for the second, so big g's, and the last one would be recessive for everything. We only have 1. So now we get this phenotypic ratio of 9 to 3 to 3 to 1, okay, and this, you will notice, might be familiar from Mendel's experiments as well, and we will see that a couple times. Okay, so that 9 to 3 to 3 to 1 ratio, keep that in mind as we move forward, but you will notice if you have 2 heterozygous parents, we should probably be getting something similar to this every time, okay? Now, we've done this first problem together, so we're going to go ahead and we're going to do another one. You can try it on your own if you would like, okay? Or we can just go through it together. So go ahead and pause here and read through this problem on your own. Okay, now that we've read through the problem, we kind of can get a sense of what we're looking at. We're going to go ahead and go through and answer this together. So, number 1, we would have to use a dihybrid cross because we are looking at 2 different genes, right, to investigate parakeet color. Okay, now this is number 2. So given the description, what are the genotypes of the 2 green parents that resulted in the white offspring? So this right here is kind of the key line, okay? Basically from this, we broke it down here and we gave you all the possible genotypes from this description. So green could be any 4 of these possible combinations, okay? Blue would have to be capital B, little y's. White has to be all recessive. The one thing it didn't mention in the problem was what if you have little b's but big y's? We can just pretend that's yellow, but it didn't really state it in the problem. So we know that the 2 green parakeets in the problem had a white offspring. So they have to be heterozygous because if they were not, there would be no possible way for us to ever get this here, right? All recessive. So our 2 green parents have to be heterozygous. Now, knowing that, we're going to go ahead and answer number 3. Okay, now we're going to draw our Punnett square, write out the phenotypic ratio. So, since both of our parents are heterozygous that we got from question 2, then we're going to go ahead and do the same thing that we did on our first practice problem. So, step 1, determine the possible gametes. So, we have that big B can go with that big Y. Okay, that's the first one. Big B could go with little y. That would be our second one. Little b could go with big Y, that is our third, and then the last one, little b with little y. Okay, that's our fourth possible gamete. Now, if this looks kind of familiar, if you're thinking, well, this looks really similar to the first one, you are correct, it does. Okay, and the reason it does is because we have 2 heterozygous parents again. Okay, so this problem is probably going to look pretty similar. Now, step 3 is to draw our Punnett square. Okay, so we got our possible gametes here. Okay, that was just from that last slide. We put out our 16 square Punnett square. We're going to go ahead and put the gametes along the top and the side. And then I want you to try to pause here and fill this in on your own. Okay, so now as you start filling in, if you have colored pencils, highlighters, okay, things that you want to kind of code this square so you remember how to do the phenotypic ratio, that's a great idea. So if you remember the dominant for both traits, okay, meaning a big B and a big Y, yellow is our dominant for one trait, recessive for the second. Okay, green is recessive for the first trait, dominant for the second, so little b, big Y, and then our last one would be recessive for both. Okay, and by doing that, we end up getting 9 to 3 to 3 to 1 again. Okay, and so again, that ratio should look familiar because if we have 2 heterozygous parents, we are always going to get this 9 to 3 to 3 to 1 ratio. Okay. So last one, step 4, it asked, question 4, it asked what was the probability that the 2 green parakeet parents had a white offspring, and that would be 1 out of 16, right? The only way that they would have that white one, okay, all recessive, so it had a 1 out of 16 probability that those 2 green parakeet parrots had a white offspring. Okay, so now that we've gone through a couple practice problems with our dihybrid crosses, you're probably thinking to yourself, okay, this is very detailed, very long. Okay, is there any other way to do this? And yes, there is. So we can also obtain outcomes of dihybrid or even trihybrid crosses without using a Punnett square. And instead we can use probabilities. So for example, in Mendel's F2 generation, which if you remember was the offspring of the F1, okay, that was the one that the F1 looked like they were all dominant, however, they were heterozygous. However, the ratio of the dominant to recessive phenotypes from that monohybrid cross was 3 to 1 or 3 out of 4. Now, how did we get that? Okay, right here, we're crossing 2 heterozygous parents, right? So heterozygous here, and we end up getting 3, okay, look dominant, and only 1 looks recessive. So 3 out of 4 look dominant. The chance of a recessive phenotype is 1 out of 4. Now, why does this matter and how can we use this? Because we can multiply. Just if you remember whenever we did our coin toss, heads and tails, okay, the chance of flipping heads 3 times in a row was a half times half times a half. We're going to do something similar here, okay? So if we multiply the chance of having yellow and round, if you remember yellow and round were both dominant traits, would be 3/4 times 3/4. Okay, and we would end up getting a 9 out of 16 chance. So let's go ahead and look, right? So Mendel's F2 generation, here was the diagram. If you remember 2 heterozygous parents, 9 to 3 to 3 to 1 ratio, if we look at this, 9 out of the 16 actually were round and yellow. Okay, so we can look at it 2 ways, either through this Punnett square and counting or simply multiplying. Okay, either one is completely fine. So same information here. We're just going to go ahead and do a couple practice problems. Okay, now, so we already did this one together, right? 2 dominant traits. Okay, now go ahead and try this one on your own. What if a plant from Mendel's F2 generation was yellow, okay, and wrinkled? What's that chance? So basically yellow is dominant, so multiply 3/4 times the recessive 1/4. If you do that, you end up getting 3 out of 16 should be yellow and wrinkled. And if you go back to that Punnett square and count, it is 3 out of 16. Okay, now last one. Now we're going to try adding a third trait. Okay, green pods is dominant over yellow, so the chance of getting big G is 3 out of 4, and little g is 1 out of 4. So now we're going to determine the probability if you have green wrinkled peas and green pods. Okay, so that green was recessive, wrinkled peas was also recessive, and green pods, which from that problem says third trait's dominant, green pods are dominant. So multiply those three together and you get there's a 3 out of 64 chance of having those three traits. Okay, now if we didn't have probability, you would have had to have drawn a 64 square Punnett square, which we do not want to do. Okay, so now we're going to do a few more practice problems together. So go ahead, pause here, read this problem, try to set this one up on your own. Okay, so if you've tried to set up that problem or do it on your own, now we're going to go ahead and go through it together. So step one, kind of break down the problem, right? So first one is heterozygous for size, but recessive for pod color. So what's your parent, okay? And the other one is recessive for size, but heterozygous for pod color, okay? So that's how we get our two parents. Now that we have our two parents, you notice that they are not both heterozygous. So this is going to be our first problem where we have different looking gametes. Okay, so first determine the gametes each parent can make. This was parent 1, this was parent 2. Now we got these combinations, so we're going to go ahead and kind of look at this together. Remember, this L could go with this G. This L could also go with this G. This little L could go with this little G. This little L could also go with this little G, and that's how we got these 4 possible combinations. The second one, this little L right here could go with this big G. That's how we get this one. This little L could go with this little G, second one. Little l, big G, this third one, and little l, little g, fourth one. Okay, so we have our 4 possible gametes from both parents. Okay, now we're going to go ahead and set up our Punnett square. Parent 1 on the top, parent 2 on the side. Okay, and then we're going to start filling it in. So go ahead and you can pause here, try to fill it in. All right, if you have filled it in, we should be getting something that looks like this, okay? Now, you notice that this one already is colored according to the phenotypic ratio down here. Again, on your paper, if you want to use highlighters, colored pencils, what have you, that is great, do that. But remember, we have dominant for both, so all of our big L's and big G's, there's these 4, okay? Dominant for the first trait, meaning a big L, little G's, 4 here. Okay, recessive for the first, little g's, little l's, big g's. Okay, recessive here for the first, dominant for the second, our green. And then all recessive, it would be our purple one, so little l's and little g's. So our ratio this time is actually 4 to 4 to 4 to 4. And if you want to reduce that to 1 to 1 to 1 to 1, perfectly fine. Okay, second check for understanding. Same thing, go ahead, pause here, and try this on your own. All right, so since we are looking at 3 traits, it says it here, you'll want to calculate probability instead of doing a Punnett square. So if you started and your first thought was, okay, 64 square Punnett square, oh, don't do that, that's going to take a really long time. So here, we're going to instead look at our probabilities. Now, both parents are heterozygous, so luckily, we already know that ratio, but just in case you forgot, here is your Punnett square showing 1 trait, okay? So essentially, if you did big H, little h on both sides, okay, you're going to get that 3 to 4 dominant looking phenotype versus 1 to 4 recessive looking phenotype. So we have those ratios down. Now, it will work the exact same for the big E little e and the big T little t, again, because they are all heterozygous, so they're all going to have the exact same ratios for this particular problem. That isn't always going to happen. Now, okay, so let's go ahead and look at the answers here, how we got them. So determine the probability if their offspring had straight hairlines, which we learned from the problem was recessive. So that's a 1 in 4 chance. If they had unattached earlobes, that is dominant, therefore 3 in 4 chance. So we multiply those two together, okay, and that would be a 3 out of 16 chance. Now we have that third trait, okay, a widow's peak, which in the problem it told us was dominant. Unattached earlobes was also dominant, and PTC tasting ability was dominant. You multiply those 3 dominant traits together, and we get a 27 out of 64 chance. Okay, Now, another similar problem, however, now we have two different parents, okay? Both parents are not heterozygous. So, step one, okay, I want you to pause here and try this, but I want you to start with a Punnett square for doing a big H, little h with two little h's, and we're going to determine the probability for those two traits, okay? So, go ahead and try that out. Pause on your here, try it on your own. All right, so step one, now that we have two different parents here, okay, we need to go ahead and do a cross to determine what the ratio for dominant to recessive is. So if we go ahead and do that here, we have 1/2 looks dominant to 1/2 looks recessive. So in this case, the chance of showing recessive phenotype is 1/2, and chance of showing a dominant phenotype is also 1/2, okay? So now whenever we multiply, we're still going to do the same thing, right? We're going to identify what the traits are, but we're always just going to end up multiplying by 1/2. So their offspring would have straight hairline. Again, that was recessive. Unattached earlobes, that was dominant. Now our chance is 1/2, again, because we have two different parents. So that's 1/2 times 1/2, and we end up with 1/4. Part B, widow's peak was dominant. Unattached earlobes was dominant, and PTC tasting ability was dominant. But now that our parents are different, again, 1/2 times 1/2 times 1/2, we have a 1 in 8 chance. So that is the end of lesson 4.3. Again, we went through a lot of practice problems in this lesson, so please make sure that you wrote down good notes, wrote out your steps, showed your work, so that way you can easily access it in class on your worksheet. Thank you for watching. Please reach out with any questions.